very clear means what are actually the meaning of selective in nature. Now, so these are literally being unstable factor means what are actually the plasma membrane. But when we are particularly understanding about the case, so you could understand by the help of the particular diagram such as we are having here. Find it. So here is the structure of the plasma membrane, or either you could say it the what the cell membrane. So it is having the different parts than the portions, such as we are having the intrinsic protein as well as the integral protein. Often what? Often the extrinsic what? Often the extrinsic protein. It will be bearing the protein channels as well as the phospholipid bilayer, as well as another portion, such as we will be having the cholesterol, as well as here made of the surface protein. Often what? Often the hydrophobic tails, or rather the hydrophobic portion, or rather the hydrophilic portion, or rather the hydrophilic part. Of course, here we will be having what? So here we will be having the phospholipid bilayer. By the name itself, it is very clear. Often it will be bearing the alpha helical protein. So you are very clear about that. Means what are actually the alpha helical protein. We'll be understanding about that later. So these are nothing else but coming under from the case of the structure of the protein. Just we have understood that what. So we have understood we are having the primary degree structure of the protein, 1 degree structure. So its function as well as the role are quite a bit different comparing to the secondary degree as well as the tertiary degree. Often these are having what? So these are having the different portions such as the cholesterol, then what? Then the phospholipid bilayer. So there was the particular scientist such as what? Such as the Jacob as well as the Nicholson. So the Jacob and Nicholson in term of the 1702 played an important role to promote as well as describe the particular structure, or either you could say that they coined the particular structure of the cell membrane, or either the plasma membrane. So it is often bearing the different parts, as well as here the particular, when we are understanding about the fluid moistening model of the plasma membrane, remember the case. So it was bearing the different portions, rather the parts, such as what? Such as we are having the intrinsic protein, then the integral protein, often the cholesterol. So each of them are literally bearing the specialized role as well as the function in it. Such as in this case we are having what? Such as in this case we are having here in term of the icebergs floating in the sea of lipids. So what does it literally means? Remember, means icebergs floating in the sea of lipid. So in this case we will be having the phospholipid bilayer and the stable factor in term present in term of the plasma membrane. Often it will be having the intrinsic protein, then the extrinsic protein. So when we are talking about the case of the extrinsic protein, so these will be literally being arranged with reference on the outer surface, or either the beneath surface, with reference to what? With reference to your phospholipid bilayer. So it could be on the right side, rather on the left side, on the upper portion, or rather the beneath portion. Likewise, but when we are talking about the case of the intrinsic protein, so this will be quite a bit specific in nature, as well as these are when particularly merged in term of what? In term of your here made of the phospholipid bilayer. So intrinsic protein will be particularly referred as the tunnel protein. So you are very clear about the structure also. Quite a bit bearing the spherical structure in terms of the tunnel protein. Quite a bit rectangular in nature. So this will be quite a bit specific in nature where they particularly emergence, or either the emergence with reference in the case of what? With reference when these are literally being implanted in the case of the phospholipid bilayer. Means in term of your what? In term of the intrinsic protein. So this will be coined as nothing else but the tunnel protein. So you are often very clear means what are actually the concentration range. So the particular portion which will be having the lower amount of the gradient, so it will be compared or either directly proportional for the lower concentration gradient, as well as the portion which is bearing the higher amount of the particles in it compared towards the another side, rather the another portion. So it will be compared as what? So it will be compared as the higher concentration gradient. Find here. So here is the lower concentration gradient, then here is the higher concentration gradient. So this is the somewhat condition when we are talking about the mechanism as well as the process of the active transport, in which we have dealt about the bit introduction, means how these processes are literally being employed, as well as what are the literally role of the carrier protein. Often we have dealt about the bit introduction, about what? About the another factors in it, such as what? Such as we have understood, means in this case the particular movement will be occurring in terms of what of the direction. Just understood means against the concentration gradient. Often we have dealt about the bit introduction, means what are actually the tunnel protein, as well as what are the intrinsic protein. Means a bit introduction means what are the mechanism of the fluid motion model, in which we have understood about the structural diagram of the plasma membrane as well as its different component and the parts role. Often we have read about the bit introduction, means what are the mechanism as well as the role of the transport protein in it, as well as how these movement are literally being employed, or either these particularly movement will be employed under what condition as well as the circumstances. Remember, so until the utilization of protein is not being done, so what will be happening? So the movement will be continued with reference to the here meant of the cell, but literally when the particular protein are utilized, the process will be submitted, or either it will be merged, or either it will be finished. So this is somewhat the condition when we are talking about the case, means what are actually the active transport. So you would often write this. So if you like the word with reference to the education as well as the lectures as well as the videos, so you could do what? So you could particularly subscribe for more lectures as well as the video as well as the content like this, as well as you could often like, then subscribe, as well as share my lectures with reference to what? With reference to the Rahul Anand biology tutorial. So very thank you.